Showing up. In 2005, Science published a list of the 25 most important questions facing the field over the next quarter century. One of those questions was, what determines species diversity? While Science acknowledged that we'd made great headway in figuring out the answers to these questions, they ultimately came to the conclusion that exactly how is largely a mystery. Now, there are a lot of great systems or examples of the role of ecology in generating new species. But these systems typically focus on how variation within a population is transformed by natural selection to differences between taxa. What's less well understood is how these changes can reverberate through trophic levels and affect entire communities or ecosystems. Now, an important but largely understudied hypothesis in evolutionary ecology is this idea that diversity begets diversity in this term that's been coined sequential or cascading divergence or speciation. Very simple concept is organisms diverge, they create new niches for other organisms to exploit and adapt to. This could potentially catalyze a chain reaction of speciation or divergence events across trophic levels. And it's been proposed that this sequential divergence process may be particularly important for explaining the incredible diversity of plant-feeding insects and the insect parasites that attack these plant-feeding insects. So in order to address this hypothesis, we use the Rhygoletes palminella sibling species complex and the parasitoids that attack these flies. These are a well-known model system for speciation with gene flow. They consist of flies, host races of flies that attack apple and hawthorn fruits, as well as a number of other taxa and named species that attack fruits of flowering dogwood, blueberry, snowberry, shrubby dogwood, and then of course things more distantly phenotypically and genetically diverged, like things that attack the black cherry. So the parasitoids that attack these flies, it's a community of Hymenopteran endoparasitic specialist wasps and the genera Eutetes, Diachasma, and Diachasmimorpha. So this is actually a genetic distance network based off of microsatellites, and it's showing populations of flies that attack different hosts across the United States. You see here flowering dogwood, followed by flies that attack apple and hawthorns, blueberry here, snowberry over here, and this is just a pattern that's showing host-associated genetic divergence. There's consistent allele frequency differences between these populations. And we see the same kind of patterns for the parasitoids that are attacking these populations, clustering in these microsatellite genetic distance networks. For all three parasitoid species, there's consistent site-to-site related allele frequency differences in these sympatric and allopatric populations that we sampled. A pattern that's consistent with the sequential divergence hypothesis. And so, in the Rhygoletes system, there are two really important ecological mechanisms that reduce gene flow between these diverging populations. One of them is that they prefer the odors emitted from the surface of their natal fruit. They avoid those odors emitted from the surface of non-natal fruits. This is results taken from flight tunnel assays just showing that flies prefer the odors emitted from the surface of their natal fruits for hawthorn flies, as well as apple flies and dogwood flies. And using wide tube olfactometer assays, we see the same results in all three species of parasitoids. The positive values represent preference for an odor. The negative values here represent avoidance for a particular odor. And across all host-associated populations for all three of these parasitoid species, we see preference for natal odor, avoidance of non-natal odor, and in almost all these cases, it's statistically significant. And this can lead to quite a bit of habitat isolation between these diverging populations of each species. So the second key ecological characteristic is these flies emerge to coincide with the fruiting phenology of the respective host plants. This spans July through September, with blueberries fruiting earlier, ranging the span all the way to flowering dogwood in September. And we see that flies emerge to coincide with the fruiting phenology of the respective host plants from blueberry, apple, snowberry, black cherry, hawthorn, and finally flowering dogwood. And the respective parasitoids that attack these flies also emerge in parallel with their fly hosts. This can lead to strong temporal isolation. We also found that there's actually microsatellite correlations with eclosion time. This graph is summarizing eclosion data across sites. So in summary, sequential speciation not only can linearly amplify biodiversity across trophic levels in a one-to-one fashion, but it seems like it's operating in a one-to-many fashion, and it may be very important for helping explain the incredible diversity of these plant-feeding insects and the parasites that attack them. We've got one example here where it's happening multiple times in a single system. I urge people that have the data to go out and look at it in their own system. Also, really cool stuff happening in this system where competition might be driving this, facilitating allochronic isolation, and could be an important driver in this system as well. And if anyone wants to talk to me about that, I'll be around.